So this is just a little video about how you can calculate genome composition. So we know in the genome that we obviously have the bases A, T, C and G are our 4 bases in DNA. So the question that we've been set here is 26% of the genome is adenine, so it's A. What percentage is guanine? Okay, so that's the question that we've been answered. So for this we have to understand the base pairing rules. Okay, so base pairs, we should know from other videos that A pairs with T with 2 hydrogen bonds, and C pairs with G with 3 hydrogen bonds. Okay, so those are the base pair rules, which means that the percentage of A must equal the percentage of T, because they're always paired together. So A will always pair with T, so you should have the same amount of A as you do T, and similarly the percentage of C must equal the percentage of G. Okay, so we can use this to work out the overall composition of the genome. So if we start with A, so we know that A is 26%. Okay, so therefore we immediately know that T must also be 26% of the genome. If it's not, then the base pairing's gone horribly wrong. Okay, so that gives us a total of 52% is A and T. Okay, so that means that C and G must equal 100 minus that, must equal 48% of the genome. Okay, so 52% is AT, therefore 48% is C and G, which must mean that C and G are half of that. Okay, so C and G must both be 48 divided by 2 equals 24% of the genome, and G is also 24% of the genome. Okay, so those should all add up to 100 because they're the only bases that we've got. Okay, so knowing the base pairing rules, it should be quite straightforward to work out the composition of the genome. And you might be thinking, oh, why do I care about it? Well, you might remember that DNA is, it's not just sort of naked DNA in the cell, it's packaged around protein. So if we've got, here's our double helix. Okay, so there's the DNA. It's not just free like that in the genome. What happens is that it ends up getting packaged. So you might have the DNA kind of then wraps around a protein called a histone. So that happens along the genome. So we wrap around again, there's a histone protein. And this is what's known as the beads on a string model. Okay, so we've got the histones, and then we actually have higher level packaging. That means that when we get to, if we think about the chromosome, what we end up having, in fact, is if this is the sort of central bit of the chromosome, I'll just put that, that's the protein kind of at the centre of the chromosome. We end up having bits of the chromosome that are, some of them are quite loosely packaged, okay, and some of them are much more tightly packaged. So we kind of end up with this loop structure, okay, of the DNA. So some bits are more highly packaged than others in what's called the chromatin. Okay? And what we know from a lot of bioinformatics, a lot of detailed studies, is that AT-rich regions are more likely to be tightly packaged, therefore are more likely to be inactive. If we're going to transcribe this DNA, if we're going to have access to the enzyme, it needs to be loosely packaged in order for transcription factors and polymerases to get anywhere near it. So if it's in this tightly packaged formation, it's likely to be inactive, and AT-rich regions tend to be tightly packaged, whereas GC-rich regions are less tightly packed, and this is on average. but regions that have quite a lot of GC in them tend to be less tightly packed, and they tend to be more biologically active. So actually, if we go down the genome, the composition of A and T might change in small regions of the genome, and that has some implications for biological function. So, but at the whole genome level, okay, we know that because of the base pairing rules, because A is pairing with T, C is pairing with G, that means that we can work out our percentages. So in this case, 26% was adenine, 26 plus 26% is 52%. That leaves us 48% for G's and C's. Split that down the middle. So what percentage is guanine? Is 24%.